Hello, I'm Scott Harper from the Center for Gene Therapy at Nationwide Children's Hospital and the Department of Pediatrics at The Ohio State University in Columbus, Ohio, in the United States. And today I'll be talking to you about gene therapy for FSHD and the efforts my lab is trying to take to develop a gene therapy approach to treat this disorder. So, to remind you from the last talk given by Dr. Saleh, the DUX4 gene is the main culprit in FSHD. That is our target. And we believe that a logical treatment for FSHD is to shut off DUX4. And that's illustrated here in this slide where we have in FSHD, DUX4 is on, the light switch is up, and the DUX4 targeted therapies that we and others are trying to develop are designed then to turn that light switch off, shut off DUX4, and hopefully then have some benefit for the disease and switch a condition from FSHD to no FSHD. And we're trying to accomplish that with an approach called gene therapy. And I'll tell you more about that in the next few slides about what gene therapy is. This is a slide from the previous talk that you may recognize. And it simply points out the idea of the central dogma of molecular biology, which is that the genetic information encoded in our genes in the form of DNA, and that's shown there at the left with the DUX4 gene, is copied into a copy of a gene called the messenger RNA through a process called transcription, and then that messenger RNA serves as a template to make protein. And the protein generally is the working body of the cell. That does the major functions of the cell. And that process is called translation when you copy a messenger RNA to a protein. So much like the previous talk, our approach is aimed at destroying the DUX4 messenger RNA. So at that center position, using a process called RNA interference, which is also abbreviated as RNAi. And a little bit different from the prior talk, we are trying to induce this RNA interference through a gene therapy strategy. And I'll tell you about that in the next slide. So what is gene therapy? It's defined as treating disease with DNA or RNA. And the concept of gene therapy has been around for many decades. Here's actually one of the first papers describing sort of theoretically what gene therapy is, way back in 1972 and published in Science magazine. And the title of this paper is Gene Therapy for Human Genetic Disease with the question mark, meaning this is all theoretical when this was first discussed. And the molecular biology field and the gene therapy fields have spent the last 40 or 50 years almost developing strategies to be able to make gene therapy a reality. And today it now is. We have approved gene therapy products on the market. So some of the features of gene therapy that are important to understand are that ideally, in a perfect situation, this should be a one-time treatment. Now, gene therapy hasn't been around long enough as an approved product to know if that's true or not, but that's how it should work. And it makes use of a viral vector to deliver the therapeutic product. So let me tell you a little bit about what a viral vector is, in the one that we are actually using for FSHD targeted gene therapy. This one is called the adeno-associated virus, also abbreviated as AAV. And these are really the viruses of choice for gene therapy. And there's several reasons for that. The AAV virus, so when I'm talking about virus, I mean the natural infectious particle, the actual virus that exists in the wild. So this AAV virus does not cause disease in humans. There are a number of different types called serotypes, and some types like to infect muscle very well. So we can use these naturally developed processes that viruses have evolved for millions of years and turn them on their head and use them for our benefit to deliver therapies to whatever target cell we want. Now, some AAV viruses don't like to hit some cells, but fortunately, like I said, they do like muscle. So upon AAV infection, these viruses deliver their own genes to host cells. But those genes do not insert into the host cell DNA. So when we deliver, or if a natural AAV virus infects you, it'll inject its DNA into your cells, but its DNA exists outside. So I've got a picture here of the host DNA, which would be like our chromosomes, and then the AAV DNA is the moon around that Earth. So the AAV DNA actually never inserts itself into our own genes, which is a good property because that has low risk of causing some sort of what's called insertional mutagenesis, which could lead to some problems. But because AAV exists outside of our DNA, but it's still stable inside our cells, it can deliver a gene product, and theoretically, at least in muscle, theoretically, it can last virtually forever. Again, that's something we still have to figure out, but it's a very long-lasting product. So that's the natural virus. What we call a vector is a modified virus. And so in this case, what an AAV vector versus an AAV virus is a little bit different, but has similar properties. So the vector system is when we take this natural virus and we remove its genes, and then we replace those natural virus genes with genes that deliver a therapy. So then we can take these modified viruses, now called vectors, and we can grow them in a lab, and they become essentially delivery vehicles for a therapy then. And these new recombinant viruses then can be delivered to cells. They're called vectors, and instead of when we deliver them to an animal model or even a human being, these vectors then inject their DNA into target cells, but instead of injecting their own DNA, the genes that are naturally present there, they're injecting this therapeutic gene. And that's essentially the concept of gene therapy. So here's an example. AAV vectors, this is an example of something my lab has done using AAV vectors. So at the top left, you can see an illustration of what we call a viral genome. So this is the viral DNA. And I just have something there called viral genes. And there are these things called ITRs. That stands for inverted terminal repeats. And within those inverted terminal repeats, any genes that are packaged within that get incorporated into the AAV particle, which is shown there in red. Now, if we cut out those viral genes and add back in some genes that we want to deliver to cells, put those inside that viral particle and then deliver those to an organism, they will deliver those genes like I told you about on the last slide. So in this example, we have a GFP gene, or GFP stands for green fluorescent protein. And this is derived, in this case, from a jellyfish. And you can see that picture of the green jellyfish there. So we can take this jellyfish GFP gene, put it inside an AAV vector, and we pick a particular vector that likes muscle. And then in this example, we've injected mice in their tibialis anterior muscle, which is essentially the muscle on the shin. And you can see that within a few weeks, that mouse's muscle that's been injected with that virus, that vector, is now glowing green, just like the jellyfish, because it's expressing that jellyfish gene. And those are the isolated muscles from that mouse. So the same concept applies when we want to deliver a therapy. And we would switch out that GFP gene for a therapy. And in this case, the main types of gene therapy that people are developing are gene replacement, gene silencing, and gene editing. Gene replacement's the best developed, and the current products that are on the market are gene replacement strategies. And what that means is that a gene is missing in a genetic disease and you add a good copy back. In our case, because our target is DUX4 and we want to shut it off, we want to develop gene silencing strategies. And that's what I'll be telling you about in the next few slides. So gene silencing is also referred to as RNAi therapy. I mentioned this RNA interference, and I'll tell you about what that is now. RNA interference, or RNAi, is a natural mechanism of gene silencing. So all of us, all human beings have about anywhere from 1,900 to maybe 2,000 natural microRNA genes. And these are just small genes, and there's one there I have located there on this slide. It's called miR30, for microRNA 30. And you can see it forms this sort of hairpin shape. These microRNAs are expressed in our genes. Like I said, that we have maybe 1,900 to 2,000 in our genes. And when they're made into these small RNAs, unlike protein coding RNAs, they don't make protein. They actually serve to regulate the expression of other genes. And what that means is, for example, this human miR30, this can exist in a cell and it can find other protein coding genes and regulate them so they don't get expressed. And this is called gene silencing. And it literally is finding a needle in a haystack. And the way this works is this microRNA has similar sequence to another gene it can bind and regulate it that way. So the important thing is that we can take these natural microRNAs and modify them for our own purposes and retarget them to silence genes that we want to knock down for example, disease treatments. And so we have developed microRNAs that can target DUX4. And so we call these microRNA shuttles. We design them with sequences to target DUX4, and they will get packaged inside a virus. We deliver that virus to a muscle cell. That muscle cell then produces these microRNAs that we've now modified inside the cell. And essentially what happens is we've made the cell, the target cell, a muscle, become a factory for producing this therapeutic product. And as long as that virus is inside that cell, this microRNA product will be made, and it can constantly produce a therapy that can target and silence DUX4. And that's the strategy. Okay, so our goal is to develop a DUX4 gene silencing strategy for FSHD that involves engineering microRNAs to silence DUX4 and then delivering these microRNAs to DUX4 expressing models and then hopefully someday to humans using AAV vectors. So our strategy for developing the FSHD gene therapy is there's basically two major components that we've been working on. The first is to actually create microRNAs that silence DUX4. And then the second is to test their effectiveness. And we've been using 3 model systems. And I'll tell you about all 3 of those in the next few slides. But the overview of it is that we have human cell lines that overproduce DUX4, and we can measure certain outcomes in those. We have human muscle cells that were donated by FSHD patients that produce natural levels of DUX4, and we can measure other outcome measures in those cells. And then we have a mouse model that we've made that produces DUX4 in its muscles and develops symptoms of muscular dystrophy. So we can test whether or not we can prevent some of those outcomes of muscular dystrophy in these mice. And I'll show you some examples of that on the next couple slides. So first of all, this is a complicated slide, and I don't want to spend too much time on it. I just want to point out a couple things, which is that at the top there on A, every one of those lines represents a microRNA that we designed and tested against DUX4. And in part C, then we have a screening procedure that I won't spend any time going over, but just to note that in this case, we've got 39 different microRNAs that we've developed against DUX4 that work really well. Our lead candidate right now, because it's effective and we've shown it's safe, is this one that we call MI405, which is shown in red and D. That's the one I'll be focusing on in the rest of the talk. Okay, so if we take these different DUX4 expressing systems and express our microRNAs in them, here are some of the things that we're looking for to demonstrate if they're effective or not. So in this first model I mentioned, we have a human cell line that we can engineer to overproduce DUX4, and if we do that, the cells die within 2 days. So the outcome measures there are DUX4 production. We can measure whether or not DUX4 is there in response to our therapy, and then whether or not those cells die. So in this case, at the top in part A, we take what's called a plasmid, which is a piece of circular DNA that expresses DUX4. We can put that inside human cells and that normally don't express DUX4, and then turn it on. And then, like I said, within 2 days, those cells will express the DUX4 protein, and if they do, then they'll die. So part B is what's called a Western blot that shows it's a way to detect DUX4 protein. And so the black band there is actually DUX4 protein. And what you can see is that that 405 sequence, our lead, compared to controls like the MIGFP, has significantly reduced DUX4 protein. So we're able to knock down DUX4 using our system, our microRNA that targets DUX4. And then the next question we can ask is, well, does this have some sort of functional effect? And one of the assays we do, one of the experiments we do is called a caspase 3,7 assay. And caspase 3,7 is just a protein that helps to initiate cell death in certain cells. And so if caspase 3,7 is high, then the cells will die. And if it's not, then the cells will live. And when we express DUX4 in these human cell line and then add in the 405 microRNA sequence, we can protect those cells from dying. So that suggested that this microRNA is protective in a cell line designed to overproduce DUX4. So we next asked, what if we put this in actual muscle cells from FSHD patients? What happens? So we have myoblasts that we acquired from other collaborators in the FSHD field. Some express DUX4, some don't. And in the center panel, then we have an example of some muscle cells from patients, and we can detect DUX4 messenger RNA in those cells. All the brown dots there, which were pointed out by red arrows in the center top, that is DUX4 messenger RNA. When we took those same cells and we expressed this microRNA, MI405, in those cells, we found very little to none DUX4 expression. And so we are able to reduce DUX4 expression in actual patient cells on a dish. And one of the things DUX4 does, it binds DNA and turns on other genes. And those genes are called DUX4 activated biomarkers. And so we also measured a couple of those DUX4 activated biomarkers in this experiment. One is called PRAM-F12 and one is called MBD3L2. And you can see the black bar represents affected FSHD cells and the normalized level of those two DUX4 biomarkers. And when we add in that 405 sequence, we're able to reduce those pretty significantly. So again, we have some benefit in FSHD endogenous cell lines, meaning that they express DUX4 naturally. And then finally, we look in mice. And in this experiment, we've taken our mouse model that we've developed that expresses FSHD or expresses DUX4 and has FSHD-like symptoms. We've packaged our microRNA inside an adeno-associated viral vector and then injected a one of these mice in one leg with the effective treatment, and then the other leg we left uninjected, or we injected it with saline. And then we let these mice age and turn on DUX4, and then we ask what the muscle looks like under a microscope. And so these pink stains are actual cross sections of muscle from a mouse in this experiment. And one of the things we do at the bottom left there, you can see a lot of blue on the right untreated leg. What we do is we go in and we intentionally color damaged muscle blue, and then we stitch it all together so you can see the extent of the muscle damage in those mice. And I think what you can appreciate is the mouse on the left, which expresses our microRNA plus this green fluorescent protein gene from jellyfish, so we can track where the virus is going, has very little of that blue damage. So we're able to protect this mouse model from DUX4-induced damage by injecting these muscles with our AAV vector expressing this microRNA. And when we quantify this, we have a pathology score, and we've done this with a number of mice. We give them a score based on the amount of muscle damage, and a score of 1 is essentially normal, score of 4 is severe muscle damage, and you can see on the MI405 treated legs, we have a pathology score approaching 1, or it is 1 in many of the animals. Again, demonstrating that we're able to protect these muscles from DUX4 damage. Finally, one of the other outcomes we do is a functional assay where we can place mice in a cage and simply monitor their activity. So we have a cage with some lasers that moves in 3 dimensions, and the mice trip these lasers as they're walking through the cage, or if they're standing up on their hind limbs. And every time it trips a laser, a computer counts that measurement. And so a mouse that has muscular dystrophy won't move as far or be able to stand up on its hind limbs as well as one that doesn't. So I think you can appreciate in this video which mouse has DUX4 on and which one does not. So we can inject mice systemically. So this means delivered through the bloodstream. So our treatment hits all muscles, and then ask if we have some benefit in this assay. And we have some data now, actually quite a bit of data now. We've taken this experiment out 10 weeks. And what you can see from this experiment is that when we deliver our treatment systemically, we can improve both activity and rearing, which is the ability to stand up on the hind limbs in DUX4 expressing mice, using two different AAV vectors, AV6 and AV9, in the blue and the red. So again, we have some global benefit from delivering this microRNA. So to summarize, gene therapy has been around for 5 decades, and though it's conceptually straightforward, it's challenging in practice. Gene therapies are getting approved for use as medical products in various countries. There are a couple of known ones, or a couple of approved ones in the United States that we know about now, and others that are getting down the path toward approval. DUX4 targeted gene therapy, we think, is a promising approach to treat FSHD. And ideally, if we do this right, an FSHD gene therapy will only require one single treatment. But because of that, we get this one shot, we need to make sure we get it done right the first time. And this requires extensive safety and dosing studies. And we think that we're getting close to human clinical trials. And I'd just like to acknowledge my lab and our collaborators and our funding sources. And I'm looking forward to the question and answer session. Thank you.